Bismillahirrahmanirrahim. Assalamu alaikum. I'm Dr. Seemal Jelani, and this fall I'm offering my course Chem 330. So, this course is basically divided into two main phases. The phase one, we will refresh some uh, information about the primary metabolites, which are carbs, proteins, lipids, enzymes, nucleic acids, and so on. First, we go for the structural analysis. We'll study the structures in detail, and after understanding the structure, then we go for the advanced biochemistry. So basically, advanced biochemistry, it is focused on the cell work, that how cell work
It's 9 o'clock.
At molecular level. So here we go for the structures and functions of molecules, and we also need to understand the enzymology, the metabolism of all these carbohydrates, lipids, proteins, and nucleic acids. Uh, we also study role of enzymes in this role of enzymes and coenzymes. We also study what is meant by the metalloproteins and enzymes, and what are the mechanisms of these enzyme actions, what are the kinetics of these enzyme actions, and what is meant by the inhibitors, and so on. So this second phase is something very we are going to dig very deeply to understand the cell work at molecular level. So as we talk about the laboratory classes, so laboratory classes in these are the classes in which we perform different experiments which are highly related to the biochemistry. We go for the first of all we go for the solution making process, how the buffer solutions are made, and then we go for the quantitative and qualitative analysis of some proteins, carbohydrates, and sugars, and after that we go for some isolation techniques. Now isolation techniques may we'll go for the isolation of casein from milk. After the isolation we go for the identification of some thing these primary metabolites, and we will go for the identification of proteins by using thin layer chromatography. Then we are going to estimate the amount of this protein by using some analytical tools, advanced analytical tools like UV absorbance, and also we go for the PCR. So this PCR, we are not going to do experiments with the PCR, but we will demonstrate you that how this PCR work. So PCR is actually, is a technique which is used for the modification of the DNA fragments or amplification of the gene fragments. So this is the course contents of this Chem 330. This is very interesting course, uh, and we learn how we can go for the research also, because by performing these little experiments, small experiments, we actually know the way by which we can conduct the different research oriented activities. So if I talk about the assessment is quizzes, is based on quizzes. Your class attendance, yes, attendance is very important, and it would be the part of assessment. And mid exam, final exam, lab activities, and final lab exams. Plus, we are going to write one academic, how to go for the academic writing, and we learn this academic writing by recording the results and everything under the heading of lab activities. So I hope you will enjoy this course in the fall, and this is very interesting course, and I hope I see you in the fall semester. Thank you so much.